Hello and welcome back. This is our fifth and final video for Chapter 6, where we will go into 6.5 Alternative Mechanisms of Carbon Fixation, where we will be looking at C4 and also KM pathways. As usual, before we go into the lesson any further, let's have a look at the learning outcomes. I hope that by the end of this video, you would be able to explain photorespiration and state the alternative mechanisms of carbon fixation done by C4 and KM plants, and also hopefully be able to do a comparison between those two alternative mechanisms of carbon fixation. So if you're ready, let's continue and have a very quick recap of what we learned in the previous subtopics. So in this chapter of photosynthesis, we went through 6.1 to 6.4, and we learned a lot about the overview, what are the components, and we learned about light-dependent reaction and also light-independent reaction, which is also called as the Calvin cycle. Now, all of this is to generate sugar molecules, but sometimes we encounter a problem here in the Calvin cycle, which is called photorespiration. Now, as a reminder to you, in case it's been a while since the previous video, photorespiration is that problem we have when we have a really high concentration of oxygen and really low concentration of carbon dioxide, and the enzyme rubisco prefers to bind to oxygen rather than carbon dioxide, and then that messes up our Calvin cycle, making it less efficient and also waste a bunch of our resources in the plants. So in this chapter, what we're going to be looking at is how in C4 plants and KM plants, these two different groups of plants came up with a way to prevent photorespiration. So kita akan sentiasa bandingkan dia dengan standard C3 plan yang kita belajar dalam subtopik sebelum ni. So dalam C3 plan yang kita belajar sebelum ni, carbon fixation berlaku sekali saja dan akan ada photorespiration bila concentration carbon dioxide terlalu rendah. Tapi dalam C4 plan dengan KM plant or CAM plant, we will see how carbon fixation will occur twice, and they will do an alternative pathway to fix carbon. So they're doing things a little bit differently so that photorespiration does not occur. And this is the main point of this subtopic. In C4 plants and KM plants, photorespiration is prevented by using an alternative mechanism of carbon fixation. In case any of you are confused, I would just like to clarify or kind of just give a very brief explanation of what is carbon fixation. Asalkan kamu nampak perkataan carbon fixation, maksudnya ada karbon dari luar dimasukkan ke dalam molekul yang sudah ada carbon. Okay, and in this chapter, we are going to focus on carbon dioxide molecules being put into something else. So kalau dia sedang cakap pasal first carbon fixation means we are taking one molecule carbon dioxide and we put it into something else. That is carbon fixation. Ada carbon dioxide, masuk. Okay, so make sure you have that concept in mind as we learn about the C4 plants. So in C4 plants, the alternative mechanism of carbon fixation is called the Hatch-Slack pathway, or you can also call it as the C4 pathway. Itu gelaran untuk mechanism dia. And examples of C4 plants are plants like corn and sugarcane. The other type of plant is KM plants. And the pathway or the mechanism that they use is the crassulacean acid metabolism or KM pathway. Examples of KM plants are cactus and pineapple. Okay, and in this subtopic, we will be looking at the C4 pathway and also the KM pathway in better detail. And here are more examples of C4 plants. So you already know about corn and sugarcane. I should also let you know that sorghum and millet plants, dua jenis tumbuhan yang juga menghasilkan bijilin, they also produce grains, are also examples of C4 plants. So let's learn more about them. Plants that follow the C4 pathway are C4 plants. it is called the C4 pathway or Hatch-Slack pathway. Kenapa kita cakap Hatch-Slack? Ini adalah merujuk kepada nama saintis yang mengkaji pathway ini. So it's a scientist with a last name Hatch, another scientist with last name Slack. Okay, so that's how we get that name. We call it C4 pathway because the first product of this pathway is a 4-carbon compound called oxaloacetate. So that's how we get the C4 name. Something that is unique to C4 plants is the structure called a Kranz anatomy. Kranz anatomy is when you have the mesophyll cells arranged in a ring form around the bundle sheath cells. And both these mesophyll cells and the bundle sheath cells will have chloroplast. So I would just like to direct your attention to this diagram over here. Kalau kamu tengok di diagram yang di bawah ni, C4 leaf ini adalah kalau kita ambil daun dan kita potong ya, we make a cross section of the leaf dan kita tengok di bawah microscope and look at it under a microscope, you will see this structure. The yellow part is the xylem and the phloem inside the leaf. This dark green cell, okay, sel-sel yang hijau gelap ini adalah bundle sheath cell yang ada chloroplast. Dikelilingi oleh mesophyll cell yang juga berwarna hijau sebab dia pun ada chloroplast. So this part here, yang bulat-bulat berlingkar-lingkar ini adalah Kranz anatomy. Now, I want to explain a little bit more about the Kranz anatomy by comparing it to a C3 leaf, which does not have the Kranz anatomy. So, ini adalah daun C3, contoh tumbuhannya adalah padi. Ini adalah C4, contoh tumbuhannya adalah jagung. Okay? So, in the daun padi, you will have these mesophyll cells in a different arrangement. The mesophyll cells, there are 2 shapes, so 2 types. The mesophyll can be palisade mesophyll, which are arranged like this up here, or the spongy mesophyll, which are arranged like this, kind of random down here. In the C3 leaf, you see this is the xylem and the phloem, and the cells that are surrounding the xylem and the phloem are the bundle sheath cells. In the C3 leaf, the bundle sheath cells are yellow to show that the bundle sheath cells in C3 leaves do not contain any chloroplast. Now we compare that to the C4 leaf. In C4 leaves, the mesophyll cells, only one shape, and the mesophyll cells will be arranged in a ring form surrounding the bundle sheath cells. So, dia bukan di atas, dia bukan di bawah. Dia akan menyelubungi, melitupi, mengelilingi bundle sheath cell. Bundle sheath cell pula memang dia akan kelilingi xylem dengan phloem. Dan dalam C4 leaf kita nampak dia punya warna hijau untuk menunjukkan bundle sheath cell dalam C4 leaf ada chloroplast. So, you can see that is the comparison between a C3 leaf that does not have any Kranz anatomy and a C4 leaf that does have Kranz anatomy. Okay, so I hope you are able to remember that. And this will be important because when we learn about C4 pathway, where it happens matters very, very much. This space and division of cells is important because in C4 plants, carbon fixation will occur twice. Okay, kita akan masukkan carbon dioxide dalam sesuatu molekul 2 kali. Pertama kali, kita akan masukkan carbon dioxide ke dalam benda lain adalah semasa kita tengok cytoplasm mesophyll cell. So, in the mesophyll cell is where we will have our first carbon fixation. And the second carbon fixation will occur in the stroma of chloroplast of bundle sheath cells. So, dalam bundle sheath cell, punya chloroplast, punya stroma adalah kali kedua di mana kita akan masukkan carbon dioxide ke dalam sesuatu. What is going to happen next is I am going to explain step by step what happens in a C4 pathway using the help of this diagram. So, kita akan berdasarkan diagram ni, kita cuba untuk tengok setiap step yang ada dalam C4 pathway. So, if you're ready, let's continue. We'll start with the first step. So this is the first carbon fixation I talked about earlier, yeah? We're going to be focusing on this region over here in the mesophyll cells. In the first step, carbon dioxide will combine with phosphoenol pyruvate, a 3-carbon compound over here. Phosphoenol pyruvate, kita pendekkan nama dia kepada PEP. So, carbon dioxide akan bergabung dengan PEP untuk membentuk oxaloacetate. So, this is the 4-carbon compound I mentioned when I talked about the name of C4 pathway. Okay, so ini adalah 4-carbon compound. And this enzyme, PEP carboxylase, will catalyze this reaction. PEP carboxylase adalah enzim yang membantu carbon dioxide bergabung dengan PEP untuk membentuk oxaloacetate. We'll talk a little bit more about this enzyme. This PEP carboxylase is an enzyme that is very faithful, sangat setia. It has high affinity for carbon dioxide. Dia suka untuk bind dengan carbon dioxide. Dan dia akan bind dengan carbon dioxide walaupun concentration carbon dioxide itu rendah. So it will bind with carbon dioxide even at low concentrations of carbon dioxide, and it will bind with carbon dioxide only. So even if oxygen enters this cell, PEP carboxylase is not going to be disturbed. Okay, so tidak akan risau dia akan tergabung dengan gas lain. Dia akan pastikan carbon dioxide akan gabung dengan PEP. So that is what happens in the first step with the help of PEP carboxylase enzyme. So then we go on to step number 2 over here. In step number 2, we'll start with oxaloacetate. Oxaloacetate will be reduced to form malate. Daripada 4 karbon, kekal 4 karbon. Reduced means it's going to accept electron. There will be reduction by NADPH, means NADPH datang, NADPH bagi elektron kepada oxaloacetate, bagi hidrogen kepada oxaloacetate. Oxaloacetate akan menjadi malate. And then NADPH becomes NADP+. What we're going to do next, now we have malate, this 4-carbon compound. Malate will be transported to the chloroplast of bundle sheath cells through the plasmodesmata. Malate akan dipindahkan kepada bundle sheath cell, masuk ke dalam chloroplast bundle sheath cell melalui plasmodesmata. Plasmodesmata are organelles that you can find on cell walls. Ataupun kamu boleh juga anggap plasmodesmata ini seakan-akan pintu-pintu di antara cell wall. Okay? It just means malate from here, go here. Itu saja maksud ayat ini. And that brings us to step number 3. Now malate is already in the bundle sheath cell. What is going to happen? Malate will undergo decarboxylation to form pyruvate. If you see the word decarboxylation, means we are removing a carboxyl group from the compound. Kita keluarkan carbon dioxide. So, bila malate, 4 karbon, kita keluarkan 1 carbon dioxide daripada malate. Malate akan menjadi pyruvate yang adalah 3 karbon. And then, we can use this carbon dioxide in whatever we want to use it for. Carbon dioxide will be released. And now, inside of this bundle sheath cells, we have high concentration of carbon dioxide gas. And this high concentration of carbon dioxide is to ensure efficient fixation by RUBP carboxylase oxygenase or Rubisco enzyme. Ingat, masalah kita sebelum ini dalam C3 plant adalah Rubisco pandai curang. Jadi, bila kita pastikan dalam bundle sheath cell cuma ada carbon dioxide, tiada oksigen, tiada benda lain mau kacau, si Rubisco ini terpaksa setia dengan carbon dioxide. And when that happens, we can make sure that we have our Kelvin cycle. So that brings us to step 4. Carbon dioxide will combine with RUBP, a 5-carbon molecule, catalyzed by the enzyme Rubisco, which now has no choice and has to bind with carbon dioxide. That will allow the Kelvin cycle to occur. forming the organic compounds in the form of G3P. So dalam diagram ini dia tunjukkan sugar, tapi kita sudah pandai kan, kita belajar subtopik sebelum ni, jadi kita tahu sugar yang dimaksudkan selepas Calvin cycle ini adalah G3P molecule. Okay, then the G3P molecule can be used however the plant wants to use it. And we need to settle something in step number 5. Okay, sudah dapat sugar, belum selesai kerja kita, kita perlu handle pyruvate. On the other hand, the pyruvate is transported back to the mesophyll cell. So daripada bundle sheet cell, kita bawa balik kepada mesophyll cell. And pyruvate will be phosphorylated to form phosphoenol pyruvate or PEP. We take phosphate group from ATP. So we hydrolyze the ATP. ATP jadi ADP. Phosphate group daripada tadi itu, kita masukkan ke dalam pyruvate. Pyruvate akan berubah menjadi PEP. And that means we can use PEP for the next carbon dioxide molecule that enters the mesophyll cell. So ini adalah kitaran ataupun aliran carbon dioxide dalam C4 pathway. So you can see once again, first carbon fixation, second carbon fixation, we make sugar, we make sure that photorespiration does not occur. So take your time to have a look at this diagram once again. And we will also do some comparison between C4 pathway and C3 pathways. So some of the differences that I can point out right now is in C4 plants, you will have two cell types that are involved. The first cell type is the mesophyll cell, and the second cell type is the bundle sheath cells. The purpose of having two cell types is for spatial separation acid, for one of them, and the second one is for sugar synthesis. So the first mesophyll cell is for creating the spatial separation acid. You have to remember here, oxaloacetate and malate are both acids. Dan dia akan pastikan dia terpisah lah. Dan sugar synthesis, which is production of G3P and glucose, will be in the bundle sheet cells. Another difference between C3 and C4 is in C4 plants, there are two carbon fixation enzymes that are involved. The first carbon fixation enzyme is RUBP carboxylase oxygenase ataupun Rubisco. The second enzyme is PEP carboxylase, which will make sure that carbon dioxide binds with PEP. Untuk ini, kalau urutan dia tidak terbalik pun tidak apa. Yang penting kamu nyatakan ada dua carbon fixation enzyme yang terlibat dalam C4 pathway. And of course, another difference is that C4 plants will have to reduce carbon dioxide twice. Or you can also say carbon fixation occurs twice in C4 plants. Okay, so these are some of the basic comparisons we can do between C3s and C4s. Of course, this explanation is not like proper explanation. Kamu perlu tahu kalau kamu mau bandingkan antara C3 dengan C4, lebih bagus kita nyatakan dalam C3 ada apa, dalam C4 ada apa. So, that is also one of the questions that they asked during PSPM many years ago in 2006 and 2007. And this is a table that quickly summarizes that. You can look at this in your own time. Look at how you can compare. Adakah kamu bandingkan berapa kali carbon fixation, siapa carbon dioxide acceptor, apa enzim yang terlibat, ada chloroplast kah, tidak chloroplast, dan sebagainya. Okay, so you can compare between C3 and C4 plants in this respect. And that's pretty much all I want to tell you about the C4 pathway. The next segment in this lesson is that I would tell you about the next alternative mechanism of carbon fixation, which is the KM pathway. And KM pathway we'll find in KM plants, which are cacti and pineapples. The KM pathway is also known as the crassulation acid metabolism pathway. Involves KM plants, of course. And this thing about KM plants, we're not going to show the diagram of the leaf, but what I can tell you is that the chem plants will not have Kranz anatomy. Okay, ini persamaan chem plant dengan C3 plant lah, dia tiada Kranz anatomy. Why we call the chem pathway as chem pathway is because we first discovered this pathway in plants that belong to family Crassulaceae. Sebab itulah family of plant yang kita duluan jumpa. That's why the name is crassulation acid metabolism pathway. And examples of chem plants are cacti and pineapples. So far, so good, right? So now I would like you to look at that image of the cactus and try to imagine what the stomata on the cactus would look like. And the answer to that is this. This diagram down here, what we have is an image of the stomata on a prickle pear cactus. So, ini adalah sejenis cactus. Kalau kamu tengok betul-betul pada diagram ini, ada part yang macam ada hijau terang ini kan? Okay, so the spots that look like bright emerald is different kind of green. That is the stomata opening of the cactus. Right now, it is open. But during the day, these stomata will be closed. So that's the thing about chem plants. So they're usually plants that live in really hot and arid conditions. So you can imagine the cactus living in a desert. During the day, it's very hot. So to prevent the plant from drying out, it's going to close its stomata during the day. To conserve water, and at the same time, it also prevents diffusion of carbon dioxide from stomata to the environment. So kalau cactus ni tutup saja dia punya stomata waktu siang, bila dia akan buka? Cactus akan buka stomata dia waktu malam. So they'll open the stomata during the night, and during the night is when you have high concentration of carbon dioxide inside the leaf due to diffusion of carbon dioxide from atmosphere through the stomata. So malam barulah carbon dioxide daripada luar akan masuk ke dalam daun ataupun ke dalam organ-organ dalam cactus dan pineapples. So what we're gonna do next is have a picture, an overall picture, an overview of what happens in the mesophyll cells of cacti or pineapples during the day and during the night. Sekarang kita tengok macam mana chem pathway dengan Calvin cycle berlaku dalam cactus. We will start with the night. Okay, so during the night time, the stoma of the plant will open. And in our imagination, in our explanation, we will be looking at the mesophyll cell of the plant only. So nighttime, stoma opens, carbon dioxide from outside can enter into the cell. So dia akan masuk dalam cell. So now we have the carbon dioxide inside of the mesophyll cell of the plant. So what will happen next is the first carbon fixation. First carbon fixation means kita ambil kita punya carbon dioxide, kita akan gabungkan dengan PEP. The same PEP that we learned about earlier. Yes. So dia akan gabung dengan PEP dan dia akan membentuk oxaloacetate. Oxaloacetate ataupun OAA, 4-carbon molecule. 4-carbon molecule oxaloacetate will then be reduced. NADPH will donate the hydrogen and the electrons, and it will become NADP+. Oxaloacetate will become malate. Malate is an acid. So we don't want this acid to lower the pH of the cytoplasm. Okay, semua ini berlaku dalam cytoplasm mesophyll cell. Jadi kita tidak mau pH dia terganggu. Kita ambil asid ini, kita letak di dalam plastik ataupun container ataupun Tupperware, dan Tupperware tersebut adalah vacuole. So we're going to transport that malate into the vacuole of the mesophyll cell to ensure that the pH of cytoplasm is not disturbed. This process that you see in this diagram over here is the chem pathway that is occurring in the cytosol of the mesophyll cell. Begitu tak? Sepanjang malam, kita akan ambil carbon dioxide dan simpan. Kita sudah bungkus, kita sudah simpan. Siang, tiba. So during the daytime, the stoma will close so that the plant does not dry out. So when the stoma closes, carbon dioxide from the outside can no longer enter into the cell. So it cannot enter into the cell. What we want to do for the Calvin cycle, okay, we still need to make sugar. So how we get the carbon dioxide from the Calvin cycle is we're going to unlock the carbon dioxide that is in the malate. So dalam malate yang masih tersimpan dalam vacuole itu, kita akan keluarkan malate. We are going to bring it to the chloroplast. Okay, so we take out the malate and we are going to do decarboxylation of malate. So kita keluarkan carbon dioxide daripada malate. Daripada 4 karbon, dia akan menjadi carbon dioxide dan molekul 3 karbon yang digelar sebagai pyruvate. So now we have the pyruvate here. We look at pyruvate later. Now we will look at carbon dioxide first. This carbon dioxide will be brought to the chloroplast and then it will bind with RUBP, catalyzed by enzyme Rubisco, and it will do the normal Calvin cycle. Normal Calvin cycle means after the cycle, it will produce sugar in the form of G3P. Okay, itu sugar, pandai-pandailah sel itu akan gunakan nanti. Sekarang kita perlu settle benda lain, yaitu pyruvate yang terhasil tadi. So, pyruvate ni kita tidak biarkan sahaja, kita perlu phosphorylate dia. So, we take a phosphate group from ATP, ATP becomes ADP. Pyruvate will be phosphorylated and becomes PEP. You see the similarity here? So this PEP will stay in the cytoplasm until we want to use it during nighttime. So ini during the day, kita kasih keluar apa yang kita simpan semalam. Kita gunakan carbon dioxide untuk hasilkan G3P ataupun glukos. Dan selebihnya pyruvate yang tertinggal itu kita akan kasih fosfat ya, supaya dia jadi balik PEP. Ada PEP, malam nanti kita boleh gunakan lagi dalam KM pathway. So that is what happens inside KM plants during the night and during the day. Now let's look at how to explain that during proper sentences. Jadi kalau kamu diminta untuk tulis essay, kamu boleh gunakan ayat yang lebih cantik ini. So during the night, stomata of KM plants will open. Carbon dioxide will combine with phosphoenol pyruvate or PEP, 3-carbon molecule, forming oxaloacetate, which is 4-carbon molecule. This reaction is catalyzed by enzyme PEP carboxylase. After that, oxaloacetate will be reduced to malate. This will be reduced by NADPH. And then malate will be stored in the vacuole of mesophyll cells. Malate is stored in the vacuole to prevent decrease in pH of the cytoplasm. And then that brings us during the day. During the day, stomata of the plant will close and malate will be transported to the chloroplast of the mesophyll cells. So dia akan dibawa keluar. And malate will undergo decarboxylation to form pyruvate. Carbon dioxide will be released, and this carbon dioxide will be used in the Calvin cycle. This carbon dioxide will combine with RUBP, catalyzed by enzyme Rubisco, and then it will continue in the Calvin cycle. Calvin cycle will occur, forming the organic compounds. Organic compounds here meaning G3P or sugars, or also glucose sugars. After that, pyruvate is transported back to the cytosol of the mesophyll cells, and pyruvate is converted to phosphoenol pyruvate, where the phosphate group will come from hydrolysis of ATP. So that is very briefly the KM pathway that we see in KM plants. I hope that you're able to remember the explanations in case you need to write an essay, and most of all, understand what is actually happening in the KM pathway. Remember, the whole point of doing this is so that there is no photorespiration that will waste our resources. And it makes sure that our photosynthesis is always efficient. So we already learned about the pathway in C4 plants and the pathway in KM plants. And as you can see here, they have some similarities and some differences. In both C4 and KM plants, carbon dioxide is incorporated into 4-carbon organic acids, or we can say it's going to have its first carbon fixation. It will become something that is 4-carbon. And also, later, that 4-carbon something or whatever organic acid is produced will release carbon dioxide to the Calvin cycle. So, kalau dalam C4 plant, this first one is over here. From CO2 becomes oxaloacetate. Oxaloacetate will later becomes malate. And malate is an organic acid. The organic acid we bring to bundle sheet cell. At bundle sheet cell, it will release carbon dioxide to do the Calvin cycle. Same thing in the KM pathway. So KM pathway, carbon dioxide enter during the night and becomes, it will combine with PEP to form oxaloacetate. Oxaloacetate becomes malate. Dua-duanya adalah organic acid. Dan bila masaknya sesuai, dia waktu siang, okay, organic acid yaitu malate tersebut, dia akan keluarkan carbon dioxide, will release carbon dioxide so that the carbon dioxide can be used in the Calvin cycle. So that is a similarity that you can see in both the C4 and the KM plants. And you can, of course, explain the similarities between C4 and KM plants in nice sentences, and then you can have a look at them here, read this in your own time so that you will be able to answer any essay questions comparing between these two types. You could also talk about the differences between these two pathways. So if you want to talk about the differences, here are some examples of differences that you can see between C4 and KM plants. So see how the categories are. Kita boleh gunakan aspect spatial separation or temporal separation of carbon fixation. Spatial separation means we do it in different places. Temporal separation means we do it at different times. Okay, and also talk about where does carbon fixation occur, which type of cell, and whether or not they have the Kranz anatomy. So that is pretty much it. Just a very quick summary. In this subtopic, we had a review of photosynthesis and also what happens in C3 plants and the problem of photorespiration. We had a look at the C4 pathway and also the KM pathway. And most importantly, you should be able to do comparison between C3 and C4, C4 and KM, and maybe even C3 and KM plants. So remember to have a look and do your own notes. And what you should do now, after all this summary, is come up with your own notes for this subtopic. In your notes, you should have a comparison table for C3, C4, and KM plants. Untuk comparison table ini, kamu boleh buat C3 dengan C4 dalam table yang sama, C4 dengan KM dalam table yang kedua. Tapi kalau kamu rasa kreatif, kamu mau gabungkan tiga mechanism of carbon fixation di dalam table yang sama, so you have a 3-column table, that's fine too. That's completely up to you as long as you come up with your own notes on how to compare between them. And in addition to that, please complete the tutorial questions for chapter 6. You should be able to answer all the questions by now and maybe even have a try at the essay questions if you would like. That's pretty much it for me. It's been quite the chapter. Thank you very much for your time and your attention, and I'll see you again soon. Happy learning and goodbye. Thank you.